All right, the last video for Unit 4 is about mitosis and cytokinesis, mostly about mitosis. Mitosis is all about making an exact copy of our genetic material to get passed into the two new cells, which are called daughter cells. This idea seems to get hit really often on the AP exam, that the purpose of mitosis is to make an exact copy, a clone of the original cell. So we put a lot of effort, of course, into making sure that our nucleus is an exact copy. Then we do need to split the cell itself, which is a little bit more haphazard in something called cytokinesis. So mitosis is in 4 steps. Whether or not you have to know these words is kind of unclear these days. It says that you don't have to, but it uses the words a lot. So prophase, metaphase, anaphase, telophase, if you can remember those, then I think that is in your best interest to try to memorize those. But knowing the events is much more important. And so, of course, we can try to just describe the events that are going on. Prophase is the first step. And so right off the bat, we have to talk about chromosomes. And so there's some vocabulary and ideas that go along with just chromosomes other than the whole like double helix thing. Most of the time in a cell, you can't see the chromosomes. Even if you use a stain that's supposed to stain the DNA is too loosely packed because it kind of looks like this, even though in this picture, this whole big old spaghetti bowl of DNA still looks like it's pretty packed. It's not that packed. And of course, the DNA strands are super, 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 super thin. But there are some ideas here that we need to introduce, such as the idea that the DNA helix is wrapped around these proteins that they're showing in gray called histones. And then those histones that are wrapped in DNA kind of wrap it on top of each other again to make something that's called chromatin. Chromatin over here. Chromatin is what we would just call the big mess of DNA in the center of the nucleus during interphase when everything's just all crazy and loose in there. But during prophase, the chromosomes need to condense. And so this picture shows a little bit more clearly that it starts to do this stuff called supercoiling. So basically, of course, the DNA double helix is a coil, which then coils around the histones, which then coil around themselves, which then keep coiling and coiling and coiling until you get this really big, thick chromosome, right? And then once they're really condensed, then you can actually see them because they're kind of thick enough to be seen with the stain. You have to put a stain on the cells, and then you can see it. And this is going to lead to some problems, though. That during the cell's life, the amount of DNA changes depending on what stage you are. Right? And so in G1, your chromosomes look like that, right? They're just long pieces of DNA. Again, they'd be more like these huge spaghetti noodles that are all like wrapped up amongst each other, but it's just easier to show them like this. But then when you get to S phase, you get DNA synthesis and you get a duplicate, an exact copy that gets stuck to it, right? And so let's just say, of course, that this is, we'll get into this more about semi-conservative replication, but there's the new one, and then here's the new one, right? So you had the left one at first, and now you have the left one and the right one. So the crazy part is that these are both called chromosomes. These are chromosomes, and these are chromosomes, but they're not equal. That's why I would call these a duplicated chromosome. Maybe you might read it as a mitotic chromosome because this is happening so that we can do mitosis, right? Now, the duplicated chromosome is made out of 2 individual pieces of DNA, but those individual pieces are called chromatids. So even though it's kind of confusing, this is a chromosome, but this is a chromatid because it's got its pair stuck to it, right? The pairs are held together at this little spot called the centromere, which technically there's a centromere all the time, but that's just where they're held together in those little spots right there, right? So that part is important, right? They're going to ask you things like how many chromatids are in this step, or tell me about how the amount of DNA changes from one step to the next. And so this is the part where you can kind of undermine yourself. by saying the word chromosome when you mean chromatid or vice versa. So once again, chromatids are the two that are stuck together. The whole thing together is called a duplicated chromosome, or sister chromatids. This is just a chromosome, right? Sometimes they'll just call that one a chromosome, but I would call it a duplicated chromosome. Right, so by the end of prophase, these duplicated chromosomes have condensed, and then we're ready to break down the nucleus, and you can see the centrioles move around, and we need these things called spindle fibers. Most of that kind of thing should be explained to you in a diagram or whatnot. The next phase is called metaphase. This is the most important phase in some ways. First, we need the spindle fibers to actually attach to each chromatid, and that's what's being shown over here on the right, on this part right here, that we have these fibers that are coming out. These are of course made out of protein, and they are going to connect right here at a thing called the kinetic core, which I will talk about in just a second. But the reason for that is so that the chromosomes can be pulled and moved around so that they will line up. The point of metaphase is to line up along the center of the cell. Sometimes it'll call it the metaphase plate. That's what this blue thing is right there. Notice that in the real pictures of cells, there is no blue plate because the plate is imaginary. But the idea is that we need our chromosomes to be lined up in more or less a straight line. Now, because this is a super important part, this is where the last checkpoint, just to remind you back to the last video, that we control the cell cycle with these checkpoints. And so the last checkpoint says, are we actually ready to separate our chromosomes? Did they line up properly? Now, I like this example. It's a little bit more detailed than you would need, but you might read about stuff like this, that the cyclins, of course, to remind us of the last video as well, are what we're actually are going to push us from one step to the next. And in the cell cycle, we're right about here, right? We are just about to be halfway done with mitosis. So if to be more precise, maybe we're right around here on the graph, right? There's this cyclin B that's hanging out. Now those kinetic cores, these things, they are sending out this signal, and that signal will stop everything from happening until it is completely attached. Once everything is nice and attached to its tubule, the chemical signal will stop, and then we can progress through the cycle. Now it turns out that the signal that gets sent out, these little, right, I just imagine little dots, little particles that are diffusing outwards, their purpose is to stop the degradation of cyclin B, right? So as soon as, here's a better way to say it maybe, as soon as these signals go away, the cell is free to break up the cyclin B, and the concentration drops really fast. And when that drops, that's the signal to go to the next phase, which would be anaphase. Again, you shouldn't have to explain it in case I did that a bad example of that. You shouldn't get into that much detail unless they would always give you the description of all this kind of thing. But I think it's an interesting way of just the variations on the theme of cyclins throughout the cell cycle. So anyway, here's where we needed to be. The cells should not start to split these sister chromatids until they are all completely lined up, because that's when like the signal is being triggered, if you want to say it that way. Then we get to anaphase. Anaphase is where you separate the sister chromatids. They move along the spindle fibers. It's kind of interesting how that happens. Again, you shouldn't have to remember this, but this is a topic that sometimes comes up. Nice diagram here to show that the little kinetochore is actually eating its way up, right? It's pulling itself this way, and as it pulls itself, it's breaking apart the tubule, the spindle fiber, and then you have all these like an exhaust. This is like the exhaust pipe of all these little protein pieces that get left in its wake. But this is how the chromosomes move around. They walk their way, eat their way up the tubule. Once they're separated enough, then we just put a new nucleus around things. This is called telophase. This is the least common thing to be talked about. It's just grouping our chromosomes together. But at the end of telophase, you end up with 2 nuclei inside the same cell, right? For a brief moment, you should have 1 cell. I know this looks like it's starting to be 2 cells in this picture over here, but maybe this would be better if this is like a dotted line. But the cell is still 1 big cell. It has 2 nuclei inside of it. Here's my little model, just to remind you that during interphase, we copy our DNA, then in prophase, they condense and the nucleus breaks down. Metaphase, they line up. I know that's not a perfect straight line, but something like that. Then they separate during anaphase, and then they group together during telophase. And here's the 2 nuclei within 1 cell, just to remind us that these 2 nuclei should be exactly the same, except for a few minor mutations that slip through. Once you have 2 nuclei, you just got to split the cell. Now, this does change depending on if you're an animal or a plant. Animals will make this fiber that kind of grows between here, and it tightens. So it's kind of being shown right in that part right there. So the tightening happens like a belt being tightened and tightened and tightened, or like if you were to tie a string around like the middle of a water balloon, if you want to think of it that way, and then you could just keep pulling the string until it was so tight that it like cut the balloon into 2 parts. Although of course the cell doesn't just like pop and all the guts spill out like a water balloon would. It's different for plants because plants have a cell wall, so they can't just like squeeze their middle part until it breaks into 2 things. Instead, they will grow a cell wall right down the center of their cell. I like that picture right here. This is the cell itself. So here's the outside kind of the cell wall, and here's the cell wall, and here's the nucleus, and then down here, the cell wall is growing. It's kind of building it as it goes. But by the end of that, whether it's in a plant or an animal, you would get 2 cells, each having a nucleus that was just newly made, which was of course made by splitting the sister chromatids, which were of course made by copying your DNA. So in the end, you get 2 cells that should be exactly the same. So there you have mitosis and cytokinesis. As always, let me know if you have any questions.